Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Hello students, welcome to the TV learning programs. As usual, I'm teacher Hurbert, who will be taking you through biology and health science for senior 3. So today we are going to be discussing unit 6, which is concerning about cell division. So today, as I told you, we're going to be discussing about the cell division, and we had to see what is cell division. Surely, I'm very happy to share with you that unit concerning about cell division, because it brings us to know how the viruses, the bacteria, the animals can multiply. Take an example, I will use an example of the COVID-19. It can just multiply and produce very many COVID-19 when they are in human or any other organism. That's why the unit we're going to discuss is concerning about cell division. Cell division. I can say when I talk about cell division, let me first ask you, what do you understand by the term a cell? Good. When I say a cell, it is a structure and it is basic structure and functional unit of the body. A cell is defined as the basic structure and functional unit of the body. So we can say the cell is the basic and structure and functional unit of the body. We can say a cell is very important because it is responsible for controlling and regulating all activities within the body. So I can say there are two types of cell division. So these two types of cell division, we have one is mitosis. Number two, we have the meiosis. So today, students, we are going to start discussing about mitosis. As I told you, mitosis and meiosis, these are two types of cell division. So take a note, and you get your exercise book, and you start making some important concepts concerning about the mitosis as a type of cell division. Let's start with the mitosis. Before I start with mitosis, you know that everyone starts as a cell, as a single cell. As you start as a single cell, because everybody starts as a single cell, are called the zygote. And then from the zygote, from the fertilization, the egg or the ovum or the sperm is joined together and form a zygote. The zygote develops into an embryo. From the embryo to the fetus. From the fetus, you become the way you exist. And I said, I remember that everyone begins as a single cell and a zygote resulting from fertilization of the egg and the sperm. This cell divides several times in order to produce more cells. We can say the cell divides several times, and after in process of dividing several times, it forms very many cells. And if they are forming very many cells, all the cells formed already, they appear from the existing cells. Okay? And that process where the cells divides from the other existing cells, that is referred as cell division. I mentioned there are two types of cell division. I said there is mitosis and meiosis. Let me start with the mitosis. Before I talk about the mitosis, I would first to explain what is inside the cell. The cell contains the nucleus. And inside the nucleus, every cell contains the nucleus. And I said the nucleus controls all the activities of the cell and regulates activities of the cell. That is to say, the nucleus contains what you call chromosomes. If I say what are chromosomes, I can say chromosomes are thread-like structures. These are thread-like structures that are found in the nucleus. Contain the chromosomes. I said chromosomes are thread-like structures. Thread-like structures that are found in the nucleus. We can also say that in the chromosomes contains what you call a DNA. If I say DNA, everybody knows what is DNA in 4. What do you understand by DNA in 4? It is referred to as deoxyribonucleic acid. And we can say one of the functions of DNA, it is a storehouse for genetic information. You can say it is a storehouse for genetic information. If I say storehouse for genetic information, that's why the DNA contains hereditary. If I say heredity, it means that is the passing of a trait or a character from the parents to the offspring. That's why some of you have a resemblance to your parents. You look alike, not exact. You look alike with your siblings or your sisters, your brothers, but not exact. That's what you passed from there. It is what you call the DNA, which is a storehouse for genetic information. I can also say that the number of chromosomes that are found in an organism or in plants vary from an organism to another organism. I can say in human, we have 46 chromosomes. I said the number of chromosomes vary from individuals, from an organism to an organism, meaning that the number of organs that are found in human are not the number of organisms that are found in lion. Let's have this one. I can say the number of chromosomes that are found in an organism, they vary according to the species. Let's say the number of chromosomes, you can say, take that note. The number of chromosomes in plants and animals, in animal cells, varies according to the species. That is say, I was saying that the number of chromosomes varies from species to species. I mean that the number of chromosomes that are found in dogs are not the number of chromosomes that are found in human beings. The number of chromosomes that are found in lion are not the number of chromosomes that are found in human. E.g., I can say in human beings has 46 chromosomes. I can say 46 chromosomes. It means that the 46 chromosomes are diploid. If I say diploid, it means that it has a complete set of chromosomes. What is a diploid cell? A diploid cell is a cell that has got a complete set of chromosomes. It's a cell that has a complete set of chromosomes. Now, that is say this one refers to be 2n. And I can say in human we have 46. That's supposed to be 46 diploid, which is 2n. Then we have haploid, supposed to be 23. This is a haploid cell. EG in Ryan, EG in Ryan, that is example 2. This is 1, and this is 2. Then in Ryans, EG in Ryans, they have 38 chromosomes, which is a diploid cell. When it is haploid, a haploid number of chromosomes that are found in the gametes of the Ryans are 19. 19 in pair. That one is supposed to be haploid. So simply, I can summarize by that one by saying that human has got 46 chromosomes, which is a diploid cell. Then a haploid number of chromosomes that are found in the gametes or in the ovum, it is 23. We have in Ryans, we have 38 diploid sets of chromosomes that are found in the ryon, but when it comes to the haploid, the gametes or the ovum are 19. Let's have the structure, the detailed structure of a chromosome. A detailed structure of a chromosome. Students, I know all of you, you must be having an idea about the structure of a chromosome. What are the major constituents of the chromosome? A detailed structure of a chromosome. Take a note and write that one. Very good. I can try to draw the structure of a chromosome. We normally say the structure of a chromosome like that. Okay? Students, I can label them with you, and you try to know the structure of chromosome. These are called chromatids. A chromatids, when it is one, it is called when it is one, it is called a chromatid. And this is where the two chromatids meet. That one is called centromere. A centromere. So let us talk now about mitosis. I said mitosis is a type of cell division. Is a type of cell division in which one cell or the mother cell divides to produce two cells or daughter cells that are genetically identical to itself. That's why I say that they are genetically identical to itself. Mitosis, take a note about mitosis, is a type of cell division in which one cell, or you can call it to be mother cell, divides, produce two new cells, or daughter cells, can call it be daughter cells, daughters that are genetically identical to itself. That is the definition of mitosis. There are other definitions. You can think about other definitions about mitosis. I said one of the definitions of mitosis, as you know, mitosis is very important in terms of the cell division. So we can also say that mitosis, the number of chromosomes in mitosis are maintained constant at the parent cell. That's why in mitosis, the offspring produced, they are genetically identical to their parents. And most of them, they are produced by means of asexual reproduction. Quickly, let's talk about the types of cell division, the types of mitosis. We must be having 5 phases of cell division. Mitosis has 5 stages. We have 5 stages. We have one is called, there are 5 phases. 5 phases of cell division of mitosis. 5 phases of mitosis. We have one which is called the interphase stage. Interphase stage. We have number 2, it's called the prophase stage. Sometimes the interphase stage is referred to as the resting phase. It's called resting phase. We have 3, it's called the metaphase stage. Metaphase stage. We have 4, is called the anaphase stage. The last, which is 5, is called the telophase stage. So students, let us go to our diagrams that can summarize what are these phases. I will start with that phase. That phase is called the interphase stage. Sometimes the interphase stage is not considered in phases of mitosis, because in interphase stage, we normally say in interphase stage, the cell is not dividing. We call it to be resting stage. But being resting stage, here there's what you call DNA replication. You can also see there's something which is called the centrosomes with the centriole pairing. We have what you call the nucleolus. We have the what you call chromation duplicated. If I say chromation, that is the process of formation of chromatins. We have the nuclear envelope. We have the plasma membrane. We have condensed chromatins. These are condensed chromatins. These condensed chromatins are the ones from what you call chromosomes. You can see from here, this is the interphase stage. We have the nuclear envelope, we have chromatin threads, we have nucleolus, we have the cytoplasm, as well as the centrioles. And we also have what you call cell surface membrane. Good. So here in that stage, we can say in that stage, what is taking place in that phase, which is called interphase stage. Let me start with the interphase stage. Interphase stage, which is referred to as resting stage in biology, resting phase. Here, what takes place there in the interphase stage, you can say there is duplication of various organelles. As I say, duplication of various organelles, it means the organelles has to multiply and become very many. So here, you can also talk about, there is also DNA replication. DNA replication. You can also say that also there is what you call active cellular metabolism. There's what you call active cellular, I can say cellular metabolism. If I say metabolism, it means there is production of more energy to be used in other stages of cell division. Let's go to this one, which is called adipophase, which is the first phase of cell division. The adipophase, you can see here, now the chromosomes, here the chromosomes are not visible, they are condensed, but here the chromosomes are visible on light microscope. And you can see the nucleus is still there. There's also chromatins, what you call intact of nuclear envelope. There's what you call centromere, where the 2 chromatids meet. We call it to be what you call centromere, attached to what you call kinetochores. We have the chromosomes. They start to appear as chromatins. They mean that here now the chromosomes are not visible, but in adipophase, the chromosomes are visible. You can also talk about this diagram. From this one is called late telophase. Late telophase. Late telophase, as you can try to see, the chromosomes are now clear, they are visible. We can say chromosomes are visible. The centrosomes move to the opposite poles of the nucleus. These are called the centrosomes, or sometimes they're called centrioles, and these are called the esters. They are moving into opposite ends of the cell. One is moving into the north pole, another one moves into the south poles. But what characterizes that? Then there is what you call the chromosomes are visible. I can say the nuclear envelope here, the nuclear envelope that was present here, you can see this is the nuclear envelope, starts to disappear. And it breaks up into smaller vesicles, which are not visible with light microscope. So these ones start to disappear. So chromosomes are seen to consist of 2 identical chromatids, and each chromatid contains 1 DNA molecule. As I told you, one of the functions of DNA, it is a storehouse for genetic information. We go to the third stage, which is called the metaphase stage. The metaphase stage, you can see the chromosomes are no longer scattered in the nucleus. The chromosomes, as you can see, the chromosomes arrange themselves along the equator of the pole. You can see what is showing the diagram is showing the chromosomes line up across the equator of the spindle, and they are attached to their centromeres. where these spindle fibers, these are called spindle fibers. These are what you call centromeres. What's something like a point in the middle of them, they're called centromeres. So the spindle fibers attaches to the centromere of each chromosome. You can see now these centrosomes, they have reached into opposite poles, meaning that the one moves into the south. As I was mentioning from here, this one has to move from south, another one moves to the north. They have reached to each pole. Each centrosome reaches the pole of the centrosomes and helps organize the production of the spindle fibers. So I can say here, the chromosomes arrange themselves along the equator of the pole. And that, when you arrange them to the equator of the pole, that one is referred as what you call metaphase plate. Metaphase plate. You can say what characterizes metaphase, the chromosomes arrange themselves along the equator of the plate. I go to the anaphase stage. Now these are, as you can see, now the chromosomes divides and forms what you call chromatids. You can see they divide. This one divides and makes a half of this. This one divides and makes a half of this. This one divides and gives a half. And they move into opposite poles. What enables to move into opposite poles? The spindle fibers enables to pull themselves into the opposite poles. You can see these are called chromatids, or sometimes they are called chromosomes now. They are no longer called chromatids, they are called chromosomes. As you can see, the chromatids move to the opposite poles. And you can say the centromeres first pull the microtubules. Now we are coming to the end of the telophase stage. The telophase stage, there is what you call now this last stage. We can say there are very many things that have to happen. Automatically, the other nuclear envelope that disappeared in the prophase stage, it has to be reformed again. You can see this one will constrict and separate and form two daughter cells which are identical to their parents. You can see this one will separate. The separation of the constriction of the cytoplasm and forms two daughter cells, that one is called cytokinesis. I can see, I said, this cytokinesis is the cell division on the cytoplasm and two biconstrictions from the edges of the cell and divides, and after dividing, totally we're going to have two daughter cells. If I'm trying to elaborate them, these are going to have one cell here and one cell here. So what characterizes that phase, there's what you call cytokinesis, the constriction and separation of the cytoplasm and form two daughter cells, and those two daughter cells must be having the equal number of chromosomes. Good. So that is what we're talking about, the mitosis. Let me try to talk about the importances. What are the significances of mitosis? I try to say the significances. What are the significances of mitosis? What is the importance of mitosis? Mitosis plays very many roles in our body. Number one, mitosis plays a role for growth. They play a role. It plays a role in growth. Everyone to grow and become an old person, it must be what we call cell division. The cell has to divide. And once they divide, you become an old person. We are born young, they become an old person. That one is done by mitosis. Number two, they are used, that is number one. Number two, replacement and repair. Replacement and repair. What does it pair? Replacement of worn-out tissues and cells are normally done by means of mitosis. Three, it is used in regeneration. If I say regeneration, that is gaining the lost parts. In some animals, mitosis plays a very big role in regeneration. That is say, regaining the lost parts. Take an example in crustaceans, an example in lizards where they can regain back their lost body parts. Take an example like sharks. The sharks normally can regain their arms and even the legs that have been lost by means of regeneration. Number four, is I can talk about what you call asexual reproduction. Asexual reproduction. So if I say asexual reproduction, that is some of the organs they don't produce by means of sexual reproduction. They produce by means of asexual reproduction, where they can produce other organisms by using asexual reproduction. So you also have to make a research and think about another one which is called genetic stability. So thank you so much for being with me in biology lesson. I hope I will be with you in the next lesson of biology. Thank you for being with me.